Hi doctor, I'm Wisin and my group members are Jian, Alyssa, Tin, Alishba and Dev. Our topic would be microbes are the future, focusing on fermented food kimchi. So why the study of fermented food is necessary and why do we choose kimchi? First is the histological and cultural reasons. Fermented food is a culinary tradition that provides insights into different cultures and diets. Fermentation is a natural preservation method. Understanding the microbial reactions in fermentations will help us to improve the preservation technique for food safety and reduces food waste. Also, fermented food contains many live beneficial microorganisms such as probiotics. Therefore, studying fermented foods allows the identifications and understanding of these beneficial bacteria, which help us to explore their potential health benefits. Lastly, fermented foods is proven to have better nutritional quality. By studying this field, we can identify the optimum fermentation conditions to maximize the nutritional value of fermented foods. Kimchi is one of the most famous Korean traditional foods and is widely consumed by people today. So it is worth studying the science of kimchi fermentations and be aware of what we have put into our bodies. This video has three parts. We will introduce the microorganisms in kimchi fermentation, their history, mechanisms, and the future prospect of the bacteria. Let's begin with the taxonomy, structures, growth conditions, characteristics, and role of the microorganisms. Lactic acid bacteria, short for LAB, is the microorganisms that initiate and drive the fermentation process. There are two types, heterolab and homolab. Heterolab produces organic acids and carbon dioxide during the early stage of fermentation. Homolab makes excessive lactic acid in the later stage of fermentation. Moving on to the taxonomy of LAB, the domain is bacteria and belongs to the Monera kingdom. Phylum, Bacillota or Firmicutes. Class of Bacilli. Order, Lactobacillus. Family, Lactobacilliaceae. Genus of Lactobacillus, and 13 more. However, we will only focus on Lactobacillus as it is the most abundant and crucial type. Finally is species. They are divided into three groups according to their metabolism. Group 1 is obligately homofermentative. Group 2 is facultatively heterofermentative. And Group 3 is obligately heterofermentative. Next is the structures of LAB. Lactobacillus is the most common rod-shaped bacteria in kimchi fermentation. It can exist in various forms such as coccus, bacillus, diploid, chain, and palisade arrangement. Then for the S layer, only some species of the lactobacilli possess a slime layer, and the majority are non-motile. Therefore, most of them lack flagella and pili. Lactobacilli form small to medium-sized gray colonies and exhibit alpha hemolysis on blood agar. Identifying the type of bacteria solely based on the morphology is less accurate. A more reliable identification method is to compare the molecular mean. In this case, lactobacilli have a 16S RNA gene sequence.
Hi, I'm Jian. Now I'll be showing the characteristics of LAB. LAB belongs to the group of gram-positive bacteria. They present as purple stain when doing Gram staining method. This is due to the presence of thick peptidoglycan cell wall. Another fact is they are catalyst-negative bacteria, means that they are responsible for breaking down hydrogen peroxide. As a result, they are unable to neutralize hydrogen peroxide toxic effects and thus setting them apart from other bacteria. Next, they are known as aerotolerant anaerobes. They can survive and grow under low oxygen or oxygen-free conditions. LAB are also non-spawn forming, which means they are more susceptible to the environment and require favorable conditions for their growth and survival. Then, LAB has no active movement as they are generally non-modal and rely on passive diffusion and external factors such as liquid flows. LAB also utilizes glucose as an energy source, converting into lactic acid, and this process known as glucose metabolism. Lastly, LAB are contamination bacterial. Although LAB has various benefits in fermentation and food production, not all bacteria in the LAB groups are desirable. In the fermentation process, undesirable flavor or spoilage can be caused by contamination of unwanted LAB strain. Next, I will be showing the growth of LAB. LAB reproduce through binary fission. It's an asexual reproduction resulting in two genetically identical daughter cells. There are 5 steps in total, which is replication, elongation, septum formation, division, and separation. First, bacterial genetic materials is replicated at the ori. Bacteria expands in the nucleic acid and its size increases. Then, a septum is found at the midpoint. Cleavage furrow separates the cells into 2 compartments. Last, the cells split and form 2 individual daughter cells. So what's the condition where LAB require for their growth? First, let's talk about temperature. The best temperature for LAB to grow is 30 to 40 degrees. The minimum growth temperature is 2 degrees and maximum is 53 degrees. Then is the pH. LAB prefer growing in acidic environment, and the best will be 5.5 to 6.2. The minimum growth pH will be around 4.5 and maximum growth pH will be around 6.5. Next, LAB reproduce in 25 to 100 minutes. There are positive and negative effects with the presence of other microorganisms. Positive effects such as synergistic and symbiotic relationship with yeast. This is because byproduct of yeast increased pH favors the growth of LAB. On the other hand, competition for nutrition is one of the negative effects, as LAB needs to compete with other microorganisms for nutrition. Then, water activity range for LAB is 0.94 to 0.99, which provides sufficient moisture to the growth of bacteria. Next, LAB as an air-tolerant anaerobe, they only need low oxygen concentration. However, LAB is weak and synthesized amino acid and vitamin, which they need high nutrition availability. And last, fermentation medium composition for LAB is rich in fermentable carbon, nucleus, amino acid, and vitamin B complex. Last, I'll be sharing the roles of LAB in fermentation. First is acidification. LAB grow readily in most food substrate, and they lower the pH rapidly to a point. This inhibits the growth of competing organisms. Next is the hydrogen peroxide production. Hydrogen peroxide produces antibiotic effects on other organisms that could cause food spoilage. Lactobacillus, one of LAB, resistant to hydrogen peroxide, which separates them from other organisms. Next is the antimicrobial action. LAB strain can produce antimicrobial compounds such as organic acid, fatty acid, peptides, and bacteriocins in fermentation food and products. This is the lactic acid production. Lactic acid can inhibit the growth of spoilage agent.
I will be talking a bit about the timeline of lactic acid bacteria and their fermentation mechanism. Did you know lactic acid bacteria are already naturally present in vegetables and environments? Traditionally, kimchi is made by natural fermentation. During fermentation, lactic acid bacteria grows in the kimchi ingredients and they give flavor and aroma of kimchi. People in the past discovered the benefits of microbial fermentation and passed the fermentation techniques to the next generation. As time went by, people began to study more about this fermentation by bacteria, and scientists will isolate the lactic acid bacteria from kimchi and then study them. Nowadays, they are using lactic acid bacteria culture in mass production of kimchi and other food industries. In addition, they could also control the conditions of lactic acid bacteria to their optimal level and improve the quality and safety of kimchi. Fermentation is a process where organic molecules, such as glucose, is converted to acids, gases, and alcohols. Fermentation occurs without the need of oxygen and they do not depend on electron transport chain. There are 2 types of fermentation, heterolytic fermentation and homolytic fermentation. In heterolytic fermentation, when glucose is converted into lactic acid, carbon dioxide, and ethanol. And in homolytic fermentation, when glucose is converted into 2 lactic acid molecules only. Both fermentation begins with the glucose entering glycolysis to give pyruvate In heterolytic fermentation, pyruvate enters pentose phosphate pathway to give cellulose phosphate, which enters phosphorylase pathway to give lactate, acetate, and ethanol, respectively. Carbon dioxide is also released during the process. And on the other hand, in homolytic fermentation, pyruvate enters anaerobic glycolysis to give lactate only. Here are examples of heterolytic fermentation: Thermobacteria species, Streptococcus species, and Leptococcus lattice. And here are examples of homolytic fermentation.
Moreover, during fermentation, lactic acid bacteria show antimicrobial activity against pathogenic bacteria. Primary metabolites such as carbon dioxide and lactic acid inhibit the growth of other unwanted bacteria, whereas secondary metabolites such as salicylic acid and azelaic acid show antifungal activity. These factors also help to get the best fermented kimchi. Low temperatures prevent the production of high amounts of lactic acid and the over-ripening of kimchi. Adding salt to kimchi also prevents the growth of non-halophilic bacteria, which is why there are high levels of salt in kimchi. Did you know that kimchi properties are also largely dependent on the metabolism of the bacteria? Lactic acid bacteria has also many health benefits on humans. For instance, they serve as probiotics for the gut, and in the gastrointestinal tract, they restore the normal intestinal flora and eliminate the intestinal pathogens. Furthermore, they aid people with lactose intolerance and malabsorption by producing lactase enzymes which break down the lactose in the food. If lactic acid bacteria are used in the combination of anti-ulcerative drugs, there will be an astonishingly fast recovery and improvement of the efficacy of treatments in patients suffering from related stomach problems. Lactic acid bacteria also has an effect on the human immune system. Staphylococcus aureus, especially if injected in its inactive state, can generate antibody immunoglobulin A. The antibodies produced can provide immunity against various antigens. Lactic acid bacteria, especially Lactobacillus citrium, can prevent the progression of antibody immunoglobulin E production that causes hypersensitivity reactions. Moreover, lactic acid bacteria also has antifungal activity. They can produce hydrogen peroxide, which has a destroying effect on fungi. Lastly, lactic acid bacteria also constitutes to a natural barrier against the mycotoxins. But on the downsides, they can also have negative effect on humans. Although they are not harmful to humans, when our immune system is weak, they can increase in number and change into infectious cells. In some people, the products of lactic acid bacterial metabolism can cause allergic reactions, although this case is rare. In the event of consuming too much kimchi or long exposure to its lactic acid, it can form an acidic environment in the oral cavity, which causes both tooth decay or enamel erosion.
I am going to be discussing the assumptions on the future prospects of LAB, otherwise known as lactic acid bacteria, in the production of kimchi. We have 4 assumptions that I am going to be discussing with you today. LAB has the potential to produce functional foods with customized characteristic profiles, such as anti-aging, anti-obesity, and antioxidative. In terms of anti-obesity, LAB plays a role in 3 different ways. Firstly, it regulates enzyme activity and inhibits microbial growth. Next, we have the assumption that as a probiotic, LAB can help balance the bacteria in our gut, which can benefit our digestive systems. They promote the growth of good bacteria while suppressing the harmful bacteria. Some studies have also shown that LAB has shown anti-cancer properties, whereby it is able to suppress the growth of tumor cells. However, there is still further research yet to be done. LAB also produces substances that can help fight against harmful microorganisms, such as bacteria, fungi, and viruses. These can prevent any infections, keeping the body in a healthy state. Next, LAB has developed improvements in biopreservation methods. Biopreservation refers to the extended shelf life and enhanced safety of food. The growth of LAB results in a preservative effect because of the reduction in pH and the competition for nutrients and the possible production of antibacterial and antifungal peptides. Lastly,
LAB possesses several characteristics that make it a promising candidate for use in the future for converting plant-derived biomass either from dedicated crops or from industrial waste into biofuel. By using LAB, we also decrease a significant reliance on fossil fuels. Because of LAB's high content and low emission rates, this provides an opportunity for bioeconomy to further grow and develop.
Hello, my name is Devindi, and I would like to talk about some of the significant challenges that bacteria in kimchi will face in the next 10 to 15 years. Microbial strain characterization. It is an important factor for the future of microorganisms in kimchi. We can better understand the unique features and functions of lactic acid bacteria found in kimchi by identifying and characterizing specific strains of it. We will be able to use this knowledge to create specialized starter cultures and optimize fermentation procedures to improve the flavor, texture, and nutritional content of kimchi. Metagenomics and metatranscriptomics are interesting approaches for studying kimchi's microbial populations. We can now analyze the genetic material and gene expression of all microorganisms in a sample using these modern techniques. We can learn about novel strains, functional genes, and metabolic pathways by unraveling the intricate relationships and metabolic activities within the kimchi microbiome. This understanding can help us develop personalized fermentation processes and probiotic compositions for greater health advantages. Maintaining a uniform standard is critical for kimchi's global acceptance and commercial success. Observing microbial populations, pH levels, and sensory qualities are all important quality control techniques to ensure a safe and desirable product. Advances in speedy and reliable analytical techniques will enable real-time monitoring of fermentation processes. While kimchi is mostly consumed as a cuisine, its applications go beyond the kitchen. Kimchi-based compounds are increasingly being investigated for their potential skincare advantages in cosmetics. However, it is critical to ensure the safety and stability of these cosmetic goods. Researchers are continuously exploring the safety of employing kimchi-derived components in cosmetic compositions, including allergenicity and stability. Addressing these concerns will pave the road for kimchi-based compounds to be used in a variety of cosmetic applications. Well, as we near the end of our presentation on microbes as the future fermented food, in which we use kimchi as our main example, let's recap the important points and analyze the importance of these discoveries. To begin, we have highlighted the widespread usage of fermented foods, with kimchi serving as a prime example because of its popularity and cultural relevance. It's an intriguing subject for research and development. Second, we emphasize the importance of lactic acid bacteria in kimchi fermentation. Both hetero-LAB and homo-LAB contribute to the fermentation process by creating organic acids and lactic acid, both of which are required for the unique flavor and preservation of kimchi. Following that, we looked at the characteristics of LAB such as the gram-positive nature, catalase negative properties, and way of reproduction by binary fission. Understanding these characteristics allow us to move effectively identify and research these bacteria. We also reviewed the beneficial benefits of LAB in kimchi, such as acidification, spoiling agent inhibition, and the formation of lactic acid and antibacterial substances. These additions not only improve the flavor and quality of kimchi, but they also contribute to its possible health advantages. We also discussed the problems involved with the future of bacteria in kimchi. These difficulties include strain characterization, quality control, metagenomics analysis, cosmetic safety issues, and overcoming the constraints of lab in fermentation. We can further optimize and improve fermentation by tackling these difficulties. Finally, the future of microorganisms in kimchi looks positive. Advances in strain characterization, metagenomics analysis, and quality control procedures will allow us to fully realize kimchi's promise. Furthermore, investigating the usage of kimchi-derived compounds in cosmetics open us to new possibilities. Let us continue to explore further into these topics as scholars and industry professionals, cooperating across disciplines and embracing innovation. We can further elevate the art of kimchi fermentation, improve its nutritional content, and broaden its numerous applications by doing so. Let us hope that kimchi will continue to pleasure our taste senses and contribute to our health in the future. Thank you.